In this talk, I want to show you an example of a research seminar that applies my research blueprint system. You might have already come across one of my other examples. For example, this altogether fictional study of a South American herb that prolongs life. Now, that example shows how the underlying blueprint always has two sides, before and after, and three levels, going from the most general or grand level to the more and more specific. If you're watching this video on YouTube, there's a link to that video in the usual information area. Now, that study was very simple, and for the fictional researchers involved, everything also went basically as expected. This time, the study will be more involved, and the going not as smooth, more like real research. The flowchart of the project I'll describe in a moment starts by following the regular pattern, moving down and then up through each of the three levels, until it gets to the updated crux level. Then, instead of proceeding to the grand level, the researchers find themselves forced to revise their original crux question, thus leading to new research activities and new outcomes at all three levels. The before portion of this project, shown here in pink, is therefore less complex than the after portion, in green. Starting with a research blueprint in this type of situation also works, and in fact it becomes even more appropriate because there is more opportunity for the complexities of the study to overwhelm the researcher's ability to present it. So, as I usually do, I will present the fictional study and then come back at the end and show you how it applies the research blueprint system. So, let me put on my hat as a research ecologist and tell you an interesting story. If you were in a plane flying from India to southern Africa and you look down about halfway, you might just be lucky enough to see this beautiful spot, the tiny island of Picobar. It's only 10 kilometres long, and it's dominated at its northern end by these two volcanic peaks. Their slopes are covered in dense rainforests, within which exists an extraordinary range of plants and animals found only on Picobar, with over 600 unique species discovered so far. Like many island ecosystems, however, Picobar is under threat. Virtually uninhabited until 50 years ago, an ever-increasing human presence is causing considerable habitat degradation, with 70 species now thought to be extinct and 20 others so threatened that they are on the IUCN's Red List. Among them is one of the flagship species of the island, the colourful Picobar tree frog. Only a decade ago, this frog could be found throughout the mid-altitude rainforests. But then its numbers started declining, and it is now estimated that the species is found across less than 10% of its previous range. My group is working to save the species from the fate of many other frogs on the island. And in the Picobar Tree Frog Reintroduction Centre, we breed large numbers of the frogs and then release them back into the wild. This is a rather inefficient and heavy-handed strategy, but we have been constrained by our limited understanding of the basic ecology of the frog. So today, I will be telling you about our recent efforts to improve that situation, and thus perhaps find better strategies for management. One important piece of information that we still don't have is the actual cause of the decline. Speculation has run along two lines. First, it's been proposed that the decline could be due to the introduction of a known predator of frogs in India, the rat snake, which was first noticed on Picobar about a decade ago. No one, however, has ever recorded a rat snake, or any other predator for that matter, actually eating a Picobar tree frog. But perhaps that's not so strange. It's quite hard to observe the frogs in the forest canopy. The second hypothesis points to the decreasing availability of an insect that is a known diet item of the Picobar tree frog. It's the Picobar lady beetle. Its own population has been decimated, also during the last decade, by a fungal pathogen. So in our research, we looked for evidence that might help us answer this question. Is the decline of the Picobar tree frog due to increased predation by rat snakes and or the decreased availability of an important diet item, the Picobar lady beetle? We started with the snakes, a seemingly less complicated matter. Now, if it is increased predation, then in the forest, clearly rat snakes must eat the Picobar tree frogs. And in captivity, in our reintroduction centre, one would predict they would also eat them. But again, this has not yet been studied. So we asked this simple question: do they? We set up 20 perspex chambers, each with a single snake in a bimodal temperature regime, as is preferred by rat snakes. The snakes were starved for a month, after which 6 frogs were placed in each chamber, 3 Picobar tree frogs and 3 brown frogs, which are about the same size as a Picobar tree frog and are a known prey item of the rat snake. The snakes quickly ate all the brown frogs, but even after a week, none of the Picobar tree frogs had been eaten. We did record some occasional attacks on the tree frogs, but the snakes quickly spat them out again. So our first experiment seemed to have a fairly clear answer: no, and thus it offered no support for our first hypothesis. Now, turning to the beetles, the situation is more subtle. Because for the decline of the beetles to affect frogs this badly, the frogs must actually depend on this one species of insect as their main source of food. This would be unusual for a frog, and it is especially so in this case because the Picobar tree frog's habitat supports over 1,400 species of insects, many of which would appear to be suitable as a food source. Now, to demonstrate a nutritional dependence of this kind experimentally is difficult, particularly so in the rather artificial setup that we use. However, one can use a proxy, an indicator for dependence. One would predict that if tree frogs were dependent on these beetles, they would, in captivity, prefer them very strongly over other insects from their habitat. So that's the experimental question that we asked. We set up another series of 20 chambers, each with a single tree frog, and after a week of starvation, 10 lady beetles were introduced into the chamber, along with 10 insects each of 4 other species found in the frog's habitat. The frogs were observed to eat the lady beetles, and, as is shown here by the large column on the left of this graph, the frogs did prefer the lady beetles. But they also ate reasonably large numbers of the other insects, including up to 50% in the case of the jasmine mayfly, that column on the right. So this is a pattern of moderate preference. It is not the pattern of strong preference that we had proposed. And as such, the data are inconsistent with the idea that the frog depends on the lady beetle as a source of food. So this was interesting. Our evidence so far was not supporting either proposal. What to do? Well, we started looking to the literature and talking to our colleagues, and this led us to an intriguing case from the forests of South America. You have probably already heard of the poison dart frogs, from which some of the native peoples extract a deadly neurological poison called a batrachotoxin alkaloid. And they use the poison on their arrows and darts for hunting and warfare. Recent evidence suggests that this toxin, which the frog is thought to use to deter predators, is derived from the consumption of certain insects called the Red beetles. And this idea is supported by experiments that have found that frogs raised in captivity on a diet free of Red beetles lack the toxin that is present in the frogs of the forest. So we wondered if a similar interrelationship might exist on Picobar, in which the lady beetles would mediate the interactions between the tree frog and its predators. And so we set up our final experiment to address this question in a captive environment. In the first stage, the tree frogs were raised on one of 5 diets, 3 of which are shown here. The percentage of lady beetles offered vis-a-vis other insects varied from 100% down to 0. 1 month later, a rat snake was introduced into each chamber for a week, after which we counted the number of frogs eaten. As this chart shows, if the percentage of beetles in the diet was 50%, rat snakes did eat some of the frogs, and ate more of them as the percentage dropped further. So, what does it all mean? Well, the results support a model in which the Picobar lady beetle does indeed play a similar role to that proposed for the South American mealy red beetle. That is, when the frogs eat the beetles, they become unpalatable to predators. Thus, the recent decimation of the lady beetle population by a fungal pathogen could have made the frogs more attractive to predators, such as the rat snake. But they also open the door, as it were, to the possible involvement of other predators, and in particular to the birds of the rainforest canopy, such as the late flying kingfisher. To put it most simply, we propose that it is the reduced availability, not of nutrition, but of a toxic defence factor, that has led to the decline of the Picobar tree frog. This, of course, is still highly speculative, but many of the predictions from this proposal are readily testable. For example, we are now studying captive frogs raised on different diets, as per our third experiment, and analysing them chemically for candidate deterrence factors. I want to conclude by returning to the question of management. The science may still have a long way to go, but given the urgency of the tree frog situation, we recommend the following. There is already a program underway to breed lady beetles that are resistant to the fungal pathogen, and we now suggest that the tree frog program should assist it with finances and other resources. Also, the removal of rat snakes, which are a pest in any case, should be given even higher priority. And finally, we encourage the bird ecologists on Picobar to focus their attention on the feeding habits of the predatory bird species that visit the canopies of the mid-altitude forests. We believe this research and that which will follow is leading us to more efficient approaches that will help to return the tree frog to its former glory in the mountain forests of Picobar. So that's the end of the fictional seminar. And now let's take a look at what's going on under the hood. Remember the basic plan of the research blueprint. 2 sides, before and after, and 3 levels: the grand level, the crux level, and the test level. In the Picobar talk, the grand level was concerned with the management of the problem. And from there, we transitioned to the more specific ecological level and our crux level question, which was the thrust of our work. To gain some insights into the ecology, we started with 2 experiments, both of which came up with answers in the negative, thus offering no support for our 2 original ecological hypotheses. Now, if that was the only research we had done, we could proceed to the implications for management directly, that is, to the grand level. That would be the classical pattern for a research blueprint. But we didn't stop there. We went back to the drawing board and we wondered whether our crux level system, that is the ecology of the Picobar tree frog, might be similar to the poison dart story, which is a real research project, by the way. And this led us to a new experiment with its own question and answer, and new implications for both ecology and, at the grand level, management. The more complex logical flow we see here is not uncommon in research, and in science at least, some high-impact journals reward researchers who carry out adventurous and difficult projects where the truth almost inevitably unfolds in a gradual and unpredictable manner. That is, not just like a story, but like an interesting story. But as complex as it seems at first, the Picobar project still adheres to the basics of the research blueprint system. It has 2 sides, before and after, and 3 levels. The grand level, which deals with major implications, the crux level, that is the point of your work, and the test level that covers your actual research activities. Now, once you develop the knack of seeing your projects in this light, you will be ready to tackle the telling of any research story. And the complexity, if it's there, instead of making your story confusing, will help to make it compelling.